Hi, this is the recording to accompany the slides for the lecture on blood. Before we start, what I thought I'd do is actually give you an overview of how these lectures are organized. So as you can see, it starts off with a title slide, followed by an overview slide over here in slide 2, and then the rest of the lecture is broken down into section. As you can see, each section is color-coded by a different color, and each section, the start of each section has got this kind of format, followed by slides with the same color with this kind of format. So you can see over here, the first section is an overview of blood, there followed by some slides to do with red blood cells, followed by other slides to do in green to do with white blood cells. And then we're going to finish off with those slides with a section on platelets over here. So when we go back and we go to the overview of the blood lecture, what we can see here is that blood is composed of 4 different components. So you've got the watery part of blood, which is plasma, followed by these 3 different sections, which are the different cells and cellular elements. So red blood cells, white blood cells, and platelets. So red blood cells are also called erythrocytes, white blood cells are also called leukocytes, and platelets, as you can see here, are also called thrombocytes. So when we talk about a blood, just as an overview, blood is considered a connective tissue. It's obviously a fluid connective tissue. And as I said, it is composed of plasma, which makes up 55% of blood. And then the other 45% is formed elements, which are cells as well as cellular elements. Blood can be either bright red when it's oxygenated or a deeper dusky red when it's deoxygenated. Blood is somewhat thicker than water to the touch, and that's because of the presence of plasma proteins and formed elements that are not found in water. In terms of the temperature, blood runs slightly higher than core body temperature due to the heat generated by friction and resistance of blood vessels as water and the different cellular form elements are transported in those blood vessels. The pH of blood runs slightly more basic than water at 7.4, and you can see here venous blood is more acidic than arterial blood due to the increased amount of CO2 in venous blood. So venous blood has an higher CO2 content, which means that it also has more protons, which then decreases the pH. And again, we'll talk more about that as we continue on. In terms of the general functions of blood, the main function of blood is transportation. So it delivers substances, it delivers nutrients to various and oxygen to various cells and tissues around the body, as well as collecting waste products such as urea and carbon dioxide in order for them to be removed from the body. Blood also has a defense mechanism in terms of the white blood cells, which help to protect the body from foreign invaders, as well as platelets, and clotting factors, which help to prevent blood loss. Blood also distributes heat. So when we're cold, what happens is blood is shunted from our superficial blood vessels in the skin deeper in the body in order to conserve heat. And when we're exercising and we're generating heat, what happens is blood moves to our superficial blood vessels in order for the heat to be dissipated. Blood is also involved in the maintenance of homeostasis, and we'll talk about this more towards the end of the course, when we talk about regulation of fluid, electrolyte, and pH balance. When we take a sample of blood and we put it in a test tube, and we put that test tube into the centrifuge, and we spin that centrifuge long enough and fast enough, what happens is it will separate out into 3 sections. So the first section is the lightest section, so it contains water and dissolved solutes. The second section, the middle section, is composed of white blood cells and platelets. And then the third section, which is the heaviest section, consists of red blood cells. So when you look at the distribution of the three different sections, what you can see here is water occupies about 55% of the volume of blood, and erythrocytes, or red blood cells, make up 99% of cells and cellular elements that are found in blood. If you look at the numbers in a unit of blood, when we measure it in microliters, you can see that there are millions of red blood cells in a microliter of blood, whereas there's only thousands of white blood cells in the same microliter of blood, and there are 100,000s of platelets in a microliter of blood. So again, most of the cells in blood are red blood cells. When we look at the composition of blood plasma, which occupies 55% of blood volume, most of this is made out of water. So 92% of blood plasma is water. The other 8% are things that are dissolved in water. Most of the things that are dissolved in water are plasma proteins, which make up 7 out of the 8% of the outstanding that's not water. And these plasma proteins are produced in the liver, and they help to maintain normal blood circulation. One of the most important plasma proteins that are produced by the liver are albumins. And albumins help to maintain osmotic balance of blood. And we'll talk more about osmotic balance of blood when we talk about the urinary system, as well as talking about fluid and electrolyte balance. Another protein that is produced by the liver and released into blood are globulins, which are an important part of the immune mechanism, as well as these plasma proteins that are important in clotting. So we'll talk about that more as we go towards the end of this lecture. There are also other regulatory substances that are present in blood, including hormones, which are transported to blood. Their hormones are produced by endocrine glands. They're released into the blood. The blood then transports them to wherever they need to go, to tissues that and cells that have receptors for the specific hormones. And growth factors, we have many different various growth factors that help different tissues to grow, as well as enzymes that help to regulate metabolism throughout the body. Other dissolved substances present in blood include ions, so positively charged ions like potassium, sodium, and calcium, as well as anions such as chloride. We also have nutrients here, as well as waste products and gases that are transported in blood. So you can see here that blood is very, very active. It's very important in maintaining homeostasis of the body. When we talk about hematopoiesis, so hemato refers to blood, poiesis refers to generation of. So what we can see here is that these blood cells are produced in a process called hematopoiesis. And what you can see here is that there are stem cells, multipotent hematopoietic stem cells, or hematocytoblast. And remember, a blast is an immature cells, and they can differentiate into a couple of different types of stem cells. So the myeloid stem cells and the lymphoid stem cells. So this happens in the bone marrow. And then what happens is the lymphoid stem cells then can differentiate into lymphoblast, and the lymphoblast then differentiate into natural killer cells, as well as T and B lymphocytes. And this happens in the lymphatic system, in lymphatic tissue, whereas the myeloid stem cells can differentiate into platelets in the end, into red blood cells, and into different kinds of white blood cells, right? So you've got the range of foreign elements over here coming from the multipotent hematopoietic stem cell. So in utero, before we're born, in the fetus, hematopoiesis occurs in the yellow sac, in the liver, in the spleen, in lymphatic tissue, and red bone marrow, whereas after birth, it is restricted to really the red bone marrow that exists within bone, as well as lymphatic tissue, especially for these lymphoid stem cells. In terms of growth factors, so these are chemicals that are released in order to regulate development of stem cells here, to precursor cells here, to mature cells over here. What happens is that there are a couple of different hormones that are produced by different organs to stimulate different types of blood cells. So the hormone that's produced that helps to stimulate red blood cell is produced by the kidneys, and it's called EPO for short, which stands for erythropoietin. And the one that helps to stimulate development of megakaryocytes into platelets is called thrombopoietin. So thrombopoietin is produced by both the liver and the kidneys, whereas erythropoietin is produced by the kidneys, right? And what happens is these two hormones regulate the development of red blood cells, as well as for EPO and platelets in case of thrombopoietin. There are also other factors called cytokines that are secreted by various other cells to help to develop these various blood cells. Just a fun fact is as well, if you are in the habit of donating your blood, what happens is plasma is replaced within 24 hours, but those cells, those red blood cells, white blood cells, platelets, take about 4 to 6 weeks to replace. So that's why the suggestion is that donating blood is awesome, but you need to actually wait a certain period in order for your cells to actually regenerate, as well as the plasma. The next section talks about erythrocytes or red blood cells. And so what you have over here is that the primary function of red blood cells is twofold. One is that it transports oxygen that we inhale from the lungs, and it transports that oxygen from the lungs to the body tissues that need it. And it also helps to transport carbon dioxide, which is a waste produced by cells and tissues, so that we can actually transport the CO2 back to the lungs so that we can actually exhale the CO2. Normally, blood has a 42 to 52% of red blood cells in men, and slightly lower for women. The difference is due to the fact that testosterone does stimulate red blood cell production. But in some cases, what happens, so this is the normal percentage of red blood cells here in a blood sample. And in the second test tube, what you have here is you have a person that is suffering from polycythemia. So if you look at that word polycythemia, poly means many, cyte refers to cells, and emia is in the blood. So someone who has polycythemia has many cells in the blood, and you may recall that 99% of cells in the blood consist of red blood cells. So someone with polycythemia has a much higher percentage of red blood cells compared to normal. And what happens is that that increase in red blood cells makes the blood thicker and makes it harder for the heart to pump the blood throughout the body. On the other hand, in test tube number 3, you've got someone who's suffering from anemia. And anemia, if you look at this, N is the absence of, emia is in the blood. So someone with anemia has a lower percentage of red blood cells in their blood. And what happens is, because the major function of blood is to transport oxygen from the lungs to the body tissues, someone with anemia has a reduced oxygen transport from the lungs to the tissue. So they can end up with a condition called hypoxemia. So hypo means a reduction, ox is oxygen, emia is in the blood. So someone with anemia, with less cells in the blood, will end up with a reduction in the amount of oxygen that's able to be transported in the blood. And this means that the ability for that person to actually do work or to walk a long distance without getting tired is reduced, right? So they can only walk a short distance before they run out of breath because they have a reduced ability to transport oxygen to various tissues in the blood. To various tissues, not various tissues in the blood. When we look at the shape and structure of erythrocytes, what we can see here is that the mature erythrocyte is shaped like a biconcave disc. Right, so a biconcave disc, when you look at it from the side, looks like this. Right, so the outer edge of the red blood cell is thicker over here, and then the middle of the red blood cell is flattened. So it's concave on both sides, right? That's what they call a biconcave thing. And what this shape allows red blood cells to do is it allows it an increase in the amount of surface area. And so you can see here is that the outside surface area of a red blood cell is much more than if it was a sphere. And what that means is that oxygen is able to go in to the red blood cell in order for the red blood cell to transport it. And then the other thing is that it allows the red blood cell to actually fold in order for it to be transported through the capillaries. And the capillaries are the smallest blood vessels that we have in the body. And what you can see here is you can see the capillaries over here, right? So this is a capillary here. And what you can see here is the red blood cell is just about the size of the capillary. And what happens is if the capillaries are a little bit smaller than the red blood cells, the red blood cell can actually fold in to be squeezed through the capillaries in single file, much like it's much easier for you to pop this donut into your mouth than it is to pop like a cream bun. So that's one thing. So that's to do with the shape of the red blood cell. When we look at what is found in a mature red blood cell, you can see here each red blood cell contains hundreds of millions of the hemoglobin protein. And because it contains so much hemoglobin, there's no room for a mature red blood cell to have any other organelles. So the mature red blood cell has no nucleus, has no ribosomes, has no endoplasmic reticulum, mitochondria, or any other organelles, because the sole purpose of the red blood cell is to contain millions of hemoglobin molecules so that oxygen can be transported, right? So it makes it a very efficient way of transporting oxygen. So what happens is that when you look over here, this is the multipotent stem cell, which then differentiates into a myeloid stem cells and then produces immature red blood cells called proerythroblast, right? And these proerythroblasts and the reticulocytes are developed in the bone marrow. And what happens is that this reticulocyte is released as an immature red blood cell into the bloodstream. And as it actually is released into the bloodstream, what happens is all the organelles in the reticulocytes actually leave or shed from the reticulocyte, so that all that remains is the hemoglobin, right? And the hemoglobin is important because it helps to transport oxygen. When we look at the life cycle of the red blood cells, what we can see here is that red blood cells are continuously being made, being produced by the bone marrow, and is continually being destroyed by the liver and spleen. And so if you look over here, you can see that the rate of red blood cell production is around 2 million red blood cells per second, and that's equivalent to the rate of destruction of red blood cells, right? So again, we need the rate of production to be equal to the rate of destruction in order for us to keep a constant amount of red blood cells in our blood. And red blood cell production is stimulated by the erythropoietin hormone that is released from the kidney. So what you can see over here is hematopoiesis begins in the bone marrow, and so it develops from a stem cell to a erythroblast to a reticulocyte. Reticulocyte is released into the bloodstream, and once they are released into the bloodstream, they take about 4 or 5 days to mature. And once they're mature, the red blood cells or the erythrocytes circulate in the bloodstream for 120 days. After around 120 days, what happens is they get old, and once they get old, they get eaten up or broken down by macrophages that are found in the bone marrow, liver, and spleen. So you can see here you've got an old red blood cell that is being swallowed by a phagocyte to be broken down by the lysosomal enzymes inside the phagocyte, so that it can be separated into its components. So it gets separated into globin amino acids and cell components. It gets separated into heme groups. It gets separated into iron. And what you can see here is that iron gets bound to the transferrin protein, and it gets recycled. And this happens in the liver, and it gets recycled in order to be used to make new red blood cells. And then the heme group gets broken down in the liver to biliverdin, and the biliverdin is broken down into bilirubin, and that is a component of bile. And we'll talk about bile later on when we get into a discussion of the digestive tract. In terms of the function of hemoglobin in red blood cells, what happens is hemoglobin is a functional protein that contains metal, and the metal it contains is iron. And so what happens is each hemoglobin protein consists of 4 polypeptide chains, so 2 beta chains and 2 alpha chains. And what happens is they combine together to form a mature hemoglobin protein. Each chain has a heme molecule that binds to a that contains an iron, and each iron can bind to 1 oxygen molecule. So you've got 4 heme proteins in a 4 heme molecules in a hemoglobin protein, each of which binds to 1 iron and 1 oxygen molecule. So each hemoglobin protein can transport up to 4 oxygen molecules. Because each red blood cells contain anywhere between 200 to 300 molecules of hemoglobin, what happens is that each hemoglobin, if you times it by 4, can transport up to 1 billion molecules of oxygen, right? So this is why it's so important for us to get the normal amount of red blood cells and hemoglobin in our blood vessels, because if we have a reduced amount of hemoglobin or reduced amount of red blood cells, it can reduce oxygen transport and cause anemia. In terms of the different types of hemoglobin, so oxyhemoglobin contains a lot of oxygen bound to it. It turns the blood bright red. Deoxyhemoglobin has less hemoglobin bound to it, so it turns the blood darker red. And carbaminohemoglobin has no effect on color. So if you look over here, you can see that both CO2 is carried in red blood cells as well as oxygen. Red blood cells are not all the same in everyone. So what happens is different red blood cells has got these different glycoproteins on them. So the glycoproteins that are most important in terms of describing a red blood cells are the A and the B glycoproteins. So someone can have, so you may know your blood type. You can be type A, type B, type AB, or type O. And that has to do with the type of glycoproteins that may be present or absent on your red blood cells. So what happens is people with type A blood has got A self-antigens on it and anti-B antibodies, right? Someone with type B blood has got B antigens on them. So you can see here, these are the B antigens. These are the A antigens on it. And because people with type B blood only have B antigens and not A antigens, they have antibodies against the A antigen. Someone with type AB blood has got both A antigens on their red blood cells as well as B antigens on their red blood cells. And because they have both types of antigens on it, they don't have any anti-A or anti-B antibodies. Someone with type O blood doesn't have A antigens, so there's no A antigens, there's no B antigens. And so what they have in their blood is antibodies to the A antigen to the B antigen, as well as antibodies to the B antigen. Right? And these antibodies help to recognize that there's not that corresponding antigen on their red blood cells, and so it helps to recognize that, for example, if you are transfused with the wrong blood type, then the antibodies can attack the antigens on the wrong blood type. So we're going to go and explain that a little bit on this. So what happens is this. So someone with, so recipient's blood is the person that's accepting the donation versus the donor's blood, which is someone who's donating the blood. So the donor's blood is given to the recipient. So if you are type O blood, what happens is you can only receive donations of type O blood, right? If you receive donations of type A, type B, or type AB blood, what happens is the antibodies in your blood will attack the antigens on the donated blood, and it will cause clumps, right? So normal blood does not clump. So you can see here you've got these individual red blood cells, whereas if you have an antibody against a foreign antigen, what happens is that the blood will clump in a process called agglutination. And what the problem with agglutination is that these clumps of blood basically block blood vessels so that red blood cells can't travel, right? So if you have these clumps of or agglutinated blood blocking the smaller red blood cells, the smaller blood vessels, what happens is then there's less blood flow in those smaller blood vessels, and then there's less oxygen being transported to those the cells and tissues that are being served by the smaller blood vessels, right? So we want to avoid agglutination. We want to avoid clumping. So what happens is, as I said, people with type O blood can only receive type O blood. People with type A blood can receive type O blood or type A blood, but cannot receive blood from someone with type B or someone with type AB, right? Because what happens is the B antibodies in this person will actually lead to clumping of the red blood cells that is donated by either someone with type B blood or someone with type AB blood. If we look at a person with type B blood, they can receive blood from type O donors or blood from type B donors, right? But not blood from people with type A or type AB. If you have someone with type AB blood, what you can see here is they can receive donations from anyone, because you don't get clumping with any of the donor, 4 donor types, right? So in this way, what we can say is that someone with type O blood can donate to all blood types, right? And that's why type O blood is in high demand, where someone with, so type O blood is universal donors, where someone with type AB blood is considered a universal recipient because they can receive blood from all the 4 major blood types. So the importance of blood typing before blood transfusion is this. So what happens when the wrong blood is transfused? So if we receive the wrong blood type, what happens is that there, on the donated blood, there are non-self antigens. And what happens is that the antibodies in your blood will actually attach randomly to the non-self blood, will end up clumping, will block small blood vessels, will cause hemolysis when the red blood cells break down. And what happens is that the kidneys can't filter the destroyed red blood cells, and the patient can very quickly develop kidney failure, right? And so we need to actually blood type before blood transfusions, or else give the person, if there's an emergency, type O negative blood, right? So in this one, what happens is there's a blood sample that's applied to these 3 different wells, and the first well has anti-A antibody, the second one has anti-B antibody, and the third one has this anti-D antibody, which is against the Rh factor. So what you can see over here is that this blood clumps in the presence of anti-A antibodies, and as well as anti-D antibodies. And so it means that this person has got the self-antigen A as well as the rhesus factor. And that's why these antibodies in these wells are clumping in there, whereas it does not have the B antigen. And so the anti-B doesn't react because there's no B antigen on the red blood cells. So the blood sample in this blood sample is type A positive. We go on next to talk about white blood cells, and there is only a short section of white blood cells because we'll talk more about white blood cells when we talk about the immune system as well as the lymphatic system. So white blood cells, as I said, is much fewer in number compared to red blood cells, so thousands compared to millions of red blood cells. They are larger than red blood cells because they are complete cells. They contain nuclei and organelles, and they can move out of blood vessels in order to actually perform their immune function. White blood cells don't last for 120 days. Their lifespan is much shorter than that of red blood cells, so they get eaten up within hours to minutes when there is an infection, right? In terms of white blood cells, they can be classified into those containing granules, so they're called granular leukocytes, and those containing less and less obvious granules, right? And so they're called agranular leukocytes. And what you can see here is that the granular leukocytes are highlighted there in yellow. They're formed in the red bone marrow as well as from the lymphoid stem line, whereas the agranular leukocytes include the monocytes as well as natural killer cells and lymphocytes. Granular leukocytes include neutrophils, eosinophils, and basophils. So they're called neutrophils, eosinophils, so they react to acid, they react to base, and this is neutral. And so what you can see over here is that neutrophils make up most of the granular leukocytes. They are the ones that are the first responders to any site of infection. And what happens is that they contain granules which have lysosomes and other enzymes for rapid destruction or phagocytosis of bacteria. Eosinophils make up less of the total number of white blood cells, and they are also effective phagocytes when there are antibody-antigen complexes being formed, whereas basophils, the granules in basophils contain histamine and other inflammatory mediators, so they help to promote inflammation, as well as containing heparin, which helps to inhibit blood clots. But again, I'm going to talk more about white blood cells when we go to the lectures about the immune system as well as the lymphatic system. As I said, white blood cells can move out of blood vessels in order to perform their immune function with infected tissues, right? So what happens is you've got these white blood cells over here within the capillaries. This is the site of injury, tissue injury, and then you've got invading pathogens here. And so what you can see here is the pathogens is quite a distance away from the blood vessels. And so what happens is when there is an injury, the injured cell releases these chemical signals, and these chemical signals are a cry for help. So the chemical signals say, hey, I'm injured. There's an injury nearby. And what happens is that chemical signals help to attract leukocytes that are passing through so that they can squeeze out of the gaps in the endothelium of the walls of the blood vessels, so they can squeeze through in a process called immigration or the diapedesis, and they follow these chemical signals to where they are most concentrated, right? Because they follow these signals to when they are most concentrated in a process called positive chemotaxis. So they're attracted positively to these chemical signals. And once they actually leave the blood vessels, then what happens is the monocytes can differentiate into macrophages, and the eosinophils and neutrophils can also help with phagocytosis to destroy the pathogens that have entered into the tissue, right? So you've got this three-step program. One is where the tissue gets injured, the injured tissue releases these chemical signals. The second part is when the white blood cells that are going through the capillaries nearby are attracted by these chemical signals in positive chemotaxis to go towards the site of injury. And then the third one is where those white blood cells actually do their job to try to destroy the pathogens that are in the injured tissue. Third type of formed elements are the platelets or thrombocytes. So you can see here, they're again developed from the myeloid stem cell that differentiate into megakaryoblast, and which then develop into megakaryocyte, which then break up into platelets, right? So each megakaryocytes releases anywhere between 2,000 to 3,000 platelets. The lifespan of platelets is 10 days, and platelets are an important part in stopping blood loss, right? So in terms of the size of the platelets, they're relatively small, and they're much fewer in number than red blood cells. Platelets are involved in a process called hemostasis. So hemo refers to blood, stasis means static, right? So hemostasis is a process that helps to prevent blood loss. So what you have here is you have when there is an injury to a blood vessel wall, what happens is then it undergoes a three-step process in sequence. So the first step is vascular spasm. So in vascular spasm, what happens is the blood vessel walls constrict, right, where there's an injury. So instead of being this wide, it constricts to try to prevent blood loss over here. And then the second step is the formation of the platelet plug. And so the platelet plug is a temporary measure in order to actually stop blood loss. And then after the platelet plug forms, what happens is that it activates clotting factors that are passing through, and those activated clotting factors then form a blood clot. So when we go to the next slide, we look at the vascular spasm, right? The vascular spasm lasts about 30 minutes or maybe hours to try to prevent blood loss. And then what happens is that when that happens, the injured cell wall then attracts platelets that are going past to actually form a platelet plug, right? So what happens is the platelets that are going past are activated by the chemicals released by the injured cells, and they become sticky and they form a clump or a plug. And the other thing that happens when a platelet plug is formed is that they release chemicals that activate and attract other platelets coming past, and those chemicals also then activate clotting factors, right? The clotting factors we're going to talk about in the next slide. The clotting factors are present in our blood all the time as inactive clotting factors. And what happens is they are ready to be activated in the case of injury, right? So we have these various different factors. I'm not expecting you to memorize all the different factors, but what happens is that they're present in blood all the time as inactive factors. And once there's an injury, the chemicals are released and they get activated, right? So there are two different pathways. There's the extrinsic pathway, where it's activated much quicker and it's more direct, activated in seconds. And the extrinsic pathway is activated when there's external trauma to blood vessel wall, right? And what happens is you've got the extrinsic pathway over here, and it leads to the common pathway, which is number 3. The intrinsic pathway is activated when there's internal damage to the blood vessel wall, right? So it's less direct, it's slower, but again, the intrinsic pathway also leads to the common pathway. And the commonality of the common pathway is the activation of this factor X or 10. When it's activated, what happens is it converts prothrombin, which is inactive, into thrombin, which is the active version. And thrombin helps to convert fibrinogen, which is a protein that's found in the blood, into fibrin. And what happens is that fibrin then forms a mesh that prevents red blood cells and other elements in blood from leaving. So you've got these fibrin threads over here actually forming a mesh similar to gauze to actually prevent platelets and erythrocytes from leaving the blood vessels, right? So it prevents blood loss. At the same time that a clot is being formed, there is also some elements that are released in the case of tissue damage that helps to start to break down clots, because a clot isn't there forever, right? A clot is there only to initially prevent the blood loss. And then what happens is when the blood vessel wall gets repaired, you don't need the clot anymore. And so that clot that is now useless needs to be broken down. And so the breaking down of clots is called clot dissolution or fibrinolysis. So it's the breakdown of those fibrin threads, right? So fibrinolysis, as it says here, is triggered by the same events that produce the clot, but fibrinolysis happens at a much slower rate than coagulation or clot formation. So when there's tissue damage, these chemicals are activated or released. And what happens is these chemicals can activate plasminogen. So again, plasminogen is present in the blood all the time as an inactive factor. And when a clot is formed, thrombin, factor 12, tissue plasminogen activator, and lysosomal enzymes can activate plasminogen and convert it to plasmin. And plasmin is the active version of plasminogen, and plasmin helps break down fibrin to actually dissolve the clot, right? And I hope you've enjoyed this. I think this is the end of